The overall goal of this procedure is to isolate adherent inflammatory leukocytes from brain blood vessels of Plasmodium berghei ANKA-infected mice. To induce experimental cerebral malaria, mice are infected with Plasmodium berghei ANKA parasitized red blood cells by injection. After the mice begin showing signs of disease, the circulating cells of malaria-infected mice are removed by intracardial perfusion. The brains are then harvested and the tissue is digested with enzymes to allow isolation of inflammatory leukocytes. Multicolor flow cytometry is then performed to immunophenotype the brain-sequestered leukocytes. Analysis of the resulting data reveals the percentage and absolute numbers of leukocytes migrating to the brain of malaria-infected mice.
This method can help answer key questions in the malaria pathogenesis field, such as what type of inflammatory cells migrate to the site of parasite sequestration in the brain of infected animals under different experimental conditions.
Visual demonstration of this method is critical. The perfusion steps are difficult to learn as they require both speed and care not to damage any significant blood vessels.
Demonstrating some parts of this procedure will be Liana McKewick's, an animal technician, Ms. Victoria Ryg-Cornejo, a PhD student, and Dr. Eliza Ioannidis, a postdoc from my laboratory.
Begin this protocol by infecting the mice with P. berghei ANKA. Defrost a cryopreserved aliquot